So on the schedule, it says it closes at 5. And if you saw my note on Monday, I have it closing at 11:59. Okay, so at 5 on Monday. So at 11. Not next Monday. Yeah. But you have to have it finished by then. You can't open, just like the regular quizzes, you can't open it before that time. It'll shut it for you. You have to open it so you have 100 minutes. And so you have to start at least 100 minutes prior to 11:59. Okay? 76. And I'll probably divide by 70. Okay, when I do that in the actual file. All right, so we're starting respiratory system. And this unit is actually respiratory and urinary. And so I have the urinary lecture notes already, but I didn't want to overwhelm you, so I didn't bring them out tonight. Your lab packet is for both. Okay? So we do have another packet for the urinary, and then after that we have digestive. So when we talk about the respiratory system, we've already talked about the major purpose of this. Yesterday, driving over from Sac State, I came up with 2 purposes, 2 functions for the respiratory system. What do you think is the first one? Pardon? To breathe. But what's the purpose of breathing then? All right, gas transport. So not only to bring oxygen to the tissues, but to carry away carbon dioxide. As we'll see, oxygen binds to hemoglobin, but most of it is dissolved in the fluid, the plasma of the blood. The other way around. Most of it's bound to hemoglobin, but we still have some dissolved in the plasma of the blood. Carbon dioxide is transported 3 different ways. It's dissolved in the blood, it's bound to hemoglobin, but most of it is carried as a form of bicarbonate, so carbonic acid or bicarbonate. Okay? But that's the purpose for both those gases, is to carry oxygen to the tissues and then carry the carbon dioxide back to the lungs so we can breathe it out. What would you think would be the second function? I'll give you a hint. If you have vomiting, so I took care of my granddaughter today because she threw up at daycare. And I didn't have, other than working on school stuff, I didn't have any responsibilities until this evening. So I went and picked her up. Now she just was vomited the one time and then she slept all day, literally. From 9 o'clock when I picked her up till 4 or 5 o'clock when I dropped her off with her other grandpa. She slept through her bottle feedings and everything else. When she opened her eyes, she was asleep again in 2 minutes. But let's say she continued to vomit throughout the day. And she lost a lot of hydrochloric acid. So that would cause the pH of her body to become more basic or more acidic. More basic, she's not reabsorbing the hydrogen ions. All right, how would the respiratory system change to compensate for that? So if she needs hydrogen ions, the other way, carbon dioxide, we'll get to this, this is part of the system. So carbon dioxide plus water forms carbonic acid. And carbonic acid is a weak acid. Some of the molecules, not all, like hydrochloric acid, but some of the molecules dissociate to form bicarbonate and hydrogen ions. So by decreasing respirations, more CO2 is available in the blood to react with water to form hydrogen ions. Okay? Now, instead, during the night, she blew out her diaper. So if instead you have a lot of diarrhea, then you're losing the bicarbonate ions from the pancreas that are functioning to neutralize the hydrogen ions from the stomach. So if you lose your bicarbonate ions, then your pH is going to be, your body pH is going to be too acidic. So in response to that, your body respiratory rate would go up, okay? And you'll be breathing off CO2, so you'll have less CO2 to form carbonic acid. Now on the reverse one, slowing down your breathing to retain carbon dioxide, there's a limit to that. We can't just stop breathing, okay? So the end of our urinary system, we're going to talk about acid-base balance. We buffer it with the respiratory system, we buffer it with the urinary system, and we use proteins and phosphates in the body to do that. But the respiratory system, yes, is primarily oxygen and carbon dioxide transport, but we use that carbon dioxide to modify it up or down to help to regulate pH. All right? Now, in this unit, we're going to talk about ventilation. That's moving. Yeah. That second thing was pH balance. Okay. So the first thing was oxygen and gas transport, and the second one was pH balance or modulation. So we're going to look at ventilation. respiration, that's moving air into and out of the lungs. Okay? So today is looking at the anatomy of the respiratory system, up to but not including the actual lungs themselves. I think you'll be tired of hearing my voice by the time we get to that point. And then we'll look at that on Thursday, and then we'll look at the mechanics of how do we change pressures to move air in and out, all right? Talking about volumes. Then external respiration is movement of the gases across the respiratory membrane. So moving oxygen across the alveolar membrane and the capillary epithelia, and moving carbon dioxide in that direction. And then internal respiration, excuse me, is the repeat of that, but with the tissues. Right? And then we'll look at the control. How does our brainstem regulate respiration? Cellular respiration we're not going to do in this unit. Cellular respiration, electron transport, and so on gets covered in digestion. Okay? So it's included, that's where we use our oxygen, and that's where we produce our carbon dioxide. But that's the specific processes we'll talk about with digestive system. All right, so there are no structures here that you need to learn for the embryonic formation of the lungs. Most of the time, what I do want you to recognize though is this first step right here, okay? So this first step involves outpouching. Do you remember our gut in our embryo folding? When we have the endoderm, and as the embryo did the longitudinal folding, it formed the central tube, and that formed the mouth and the esophagus, and we get to the digestive system, it forms stomach, and outgrowths form the liver and the pancreas. So as we first have our mouth area, or our somatodeum, and that's where we talked about the outgrowth for the anterior pituitary from the pharynx. And then this comes down to form more of the pharynx, and then out of that, we have a growth that is for the trachea and the rest of the lung tissue. Most of this division is dichotomous, branches into two. So we see the trachea eventually is going to branch into the two bronchi buds, one for the right lung, one for the left. And then in an unusual situation, instead of having continuous dichotomous branching, the right forms three branches. We have three lobes of the lung, and the left forms two. Okay? After that, we continue with our process of primarily dichotomous branching. So the author of the embryology book that we saw our, okay, what happened to you here? I'm gonna go back. That we saw our folding, Keith Moore, has a short little video for the embryonic development of the lungs. So let's just look at that. And again, there's no names or structures or anything that you need to know here. I had this set up, but already today we did add. Okay, so here's the embryo. So we see liver and pancreas folding there, and this is the first outpouching from the pharyngeal area that we're going to see. These are called pharyngeal or brachial arches that form certain structures in the body. Heart's developing here. And I'm going to skip forward a little bit because it spends a lot of time on those pharyngeal structures. We're looking at the throat. Where's the front but from the back? You're seeing the back of the tongue. This is where the little tonsils are developing. Retinoids are a pair of cartilages that sit on the top of the posterior view of the larynx. So the lining of the respiratory tract is going to develop from the endoderm that we saw in the embryonic development. non-fortified stratified squamous. We'll talk about that as we talk about the structures. And the trachea is going to go back to the pseudostratified columnar that we see in the lineal cavity. And remember, in the embryo, as the lung is developing, there's no air available, right? So we're not doing any gas exchange. The purpose, the alveoli, where we normally would have filled with air after birth, are filled with fluid. And that's to keep them from developing as solid tissue, because that's not going to occur with the first breath that the infant takes. It has to already be there. So this fluid starts to be reabsorbed several weeks before birth. One of the issues with premature infants. Not only have they not started making the oily substance that coats it, but they haven't started that reabsorption. Additional fluid is expressed out with birth through vaginal delivery, and again, C-section babies are more at risk for having fluid in their lungs, even if they are mature. Okay? So here you can see the 3 branches for the 3 lobes of the right lung, the 2 branches for the 2 lobes of the left lung. That's thought for the same reason we have 2 atrioventricular valves in the heart, to be because of the movement of the heart is more on the left side, so there's less room for development on the left side. So 5th week, you don't have to know the dates. Beginning of the 5th week is only 2 weeks after the heart starts to beat. So we already have fluid, plasma, blood circulating through this tissue. So the yellow is the epithelial lining. And then outside of that, we have the smooth muscle structures, all right? And blood vessels and so on. Actually, the yellow is the spaces, and this deeper red here is the epithelial lining. So here we have the epithelial lining, blood vessels, and connective tissue. That's from the splanchnic mesenchyme. So we don't have any skeletal muscle down that area, but we have smooth muscle around the airways as well as around the blood vessels. Okay? All right. So now let's talk about the organization of the airway. And I don't know if you remember in Bio 430, when the nervous system was discussed, we organized the nervous system into central and peripheral nervous system. That was by location. Okay? And then we also described it as motor and sensory, and that was by function. So we have the same 2 types of organization for the respiratory system. Conducting zone and respiratory zone is by function. All right? As we'll see towards the end of the lecture tonight, when we get to the respiratory zone, this involved the addition of the tissue that allows gas exchange. So as we think about back in, again, Bio 430, when we first learned about epithelial tissues, when we were looking at functions, we had a lot of room in our cuboidal and columnar cells for Golgi apparatus and ribosomal ER for protein synthesis, all right? Stratified layers were for protection against absorption or abrasion. So on the skin, we didn't want to become dehydrated. We saw it in our last unit as a mechanical barrier for pathogens moving through. And then our simple squamous epithelium was thin enough that it didn't secrete much, maybe a watery serous fluid, all right? We'll see in the urinary system where we use it for filtration, when we're separating particles by size from the blood into the blood versus the filtrate. And it's also very thin. We see it in the capillaries, right? So in the capillaries, it's there to allow the movement of nutrients, including gases, into waste products, into and out of the blood. So, which of these epithelia would have to be present for gas exchange to occur to classify a structure as respiratory zone? Because if we have a respiratory zone, we're exchanging gases. All right, we have to have epithelia that allows that gas exchange to occur. Now, as we move through different from the trachea and the bronchus because it lacks cartilage. So there's nothing to prop the airway open and maintain it open like it's did. It has cilia, and it can still secrete mucus. But that tissue right there is still too thick for gas exchange, especially with the presence of the smooth muscle. But the epithelia itself is a simple cuboidal, is too thick for gas exchange. So we start to see an occasional bleb, like a grape, that is a single alveolus all by itself popping out of a bronchiole. All right? There's still smooth muscle present, so the bronchiole is going to continue. But all of a sudden, we have this alveolus, all right, with simple squamous epithelia, and if we have capillaries on the other side of it, also with simple squamous epithelia. Notice the nucleus was on the opposite side of the airway, so that we have a narrow distance for gas exchange. So as soon as this first alveolus appears, the rest of this airway is identified as a respiratory bronchiole. Okay? So we'll go back to the previous slide. We're going to talk about this like about probably 15 minutes before class is over this evening, maybe 20. And so if we go back to the previous slide on functional classification, you'll see that the respiratory bronchiole is the first airway that fits into the respiratory zone, because that's the first time we have gas exchange. All right? So we describe the histology of all these airways later on this evening. But that's why it's called a respiratory bronchiole. It has the structure of a bronchiole, but then it has alveoli within it as well, allowing the gas exchange. So all the structures after that have at least something, if not everything, as part of the airway that contains the thinnest epithelia that you see up here. All right? So we want the single layer and the thinnest. So then the second mechanism of classifying airways is by location. You can see in the upper right there it says lower respiratory zone, and that's starting with the trachea. Right? Upper respiratory tract is the upper half of the anatomical separation. So it says zone up there. Tract is what I tend to use. And this is anatomical separation, not functional. It's going to include the nasal cavity, all of the parts of the pharynx. So the nasopharynx, laryngopharynx, and oropharynx that we're going to talk about, and the larynx. Okay? That's upper respiratory tract. The lower respiratory tract will start at the distal end of the larynx with the trachea, and will include everything down to the smallest airway passage in the lungs. So, if you have laryngitis, or tonsillitis, or cold, all right, then you have an upper respiratory tract infection. If you have bronchitis, or pneumonia, or emphysema, then you have a lower respiratory tract condition. So here's the larynx. So anything above that would be upper respiratory tract. Anything below that would be lower respiratory tract. Okay? Now, depending on the textbook, some will include the larynx in the lower respiratory tract. So we're just going to go with the group that goes below the larynx. Okay, it's arbitrary, but that's where we're starting with this group. Okay? So as we look at this, you can see the trachea has cartilage. The larynx is made of cartilage, so the trachea is not the first structure. It helps to maintain the airway. The bronchi continue to have cartilage. And then when we get to bronchioles, they no longer have cartilage. All right, so see, the first time we see respiratory bronchial, these little grape-like structures are what I drew on the board. And these are the structures, the little alveoli, that we have gas exchange. And finally, that's all we have, are just clusters of alveoli. Okay? So in that lower respiratory tract, you can also see the functional change. All right, so I'm going to diagram this on the board, and we're going to talk about, do a little review of structures and histology. But essentially what we're looking at here is a diagram of the sagittal view of the head. All right, so we're going to come back to the oral area and oral pharynx when we do digestive system. But we're going to start here with the air passageway into the external nares. So this is what I'm going to diagram on the board. If you have a hard time following the diagram, you can look at the image, okay? But what I'm going to add to it are some of the histology and bone structure reviews that we haven't covered for a while, which reminds me, let me grab this model back here. This is a new model. I did not have this available last year. And it makes some of our definitions a lot easier for students to understand. We do have a quiz on Thursday. The quiz is going to be primarily over what you can see up here. All right, well, not primarily. Everything that we see up here, including the larynx. It will not include the histology. All right, we'll have for both urinary system and respiratory system. We'll have a quiz on anatomy, structures, models and diagrams, and then we'll have a separate quiz on the histology of those areas. All right. Body. And that is the nose. All right. The opening of each nostril is called a nares. We have an internal set and an external set of nares. So this is known as an external nares. Two nares together, plural. We change IS to ES. Okay? So if we have air passing into that region, essentially this part right here that you can collapse with your fingers is known as the vestibule of the nostril. So the whole area here is the nostril, but the space of that is the vestibule. Okay? It has a distinctively different epithelia than the rest of the nasal cavity. This is the same epithelia that we find in the oral area, and that's because air can be drying. And so to protect that area, we have wet surfaces deeper. But to protect that area, this is going to be, not is, but non-cornified, or if you're more familiar with non-stratified. Non-cornified, non-keratinized, non-cornified, the same. Non-cornified, stratified, squamous. So the same type of epithelium that we've discussed lining the vaginal region, lining the mouth, that will line the throat, and so on. All right? Then we enter the nasal cavity proper. And the nasal cavity is separated from the mouth by two bones. So if you take your tongue and reach up and touch the roof of your mouth, the bone that you're touching would be what bone? Pardon? Not the palatine bone. It is the palate, but it's the palatine plate or the horizontal plate of the same bone out here. Maxillary bone. Okay, or maxilla. So this would be the maxilla. And The posterior third of the palate is the palatine bone, okay? So if you were to turn the skull upside down, the arch of the tooth, so if we look at this just briefly, this is maxillary, and this is palatine, okay? I just don't think your tongue could reach that far back. So that will be the palatine. I'm just going to put a P, A-L. And that's the hard palate, all right? So that's going to be covered with epithelia. And then that is going to hang down in the back as the soft palate. And that's when we look at it in the mouth, and we'll talk about this more when we do digestive system. So hanging from the soft palate is the uvula. I'm not going to require you to know that for this unit, but that's the little finger-like structure hanging down from the palate in the back of your mouth. But that's just in the center. The rest of this doesn't have any bone and moves up and down with speech and with coughing. Okay, so separate nasopharynx from oropharynx. All right, so what's the bone forming most of the roof of the nasal cavity? We have the nasal bone here. So up here we have,
I have a quick question.
Yeah.
Uh-huh. So the soft palate is the palatine bone?
no, soft palate is where we have no bone. So if you look on the diagram here, here's maxillary, here's palatine, and then we have an area of muscle and epithelia that has no bone. Yes. Yeah. Okay. So most of this up here is frontal bone. But then we have a region in here contributing to the crista galli, and there are pores passing through that allow olfactory neurons to send their axons, okay? And that's called the cribriform plate. What bone is that? What bone has the cribriform plate? Not the sphenoid. Sphenoid is going to be back here, where we have the sphenoid sinus. Pardon? Hyoid? No. Ethmoid. Okay, we have a lot of processes in the nasal cavity from the ethmoid bone. So I'm going to write it separately in green like I did here, because we have more of our structures. So this is going to be the cribriform plate. I'll put ethmoid bone first. And we have the cribriform plate of the ethmoid bone for the olfactory neurons. And then we have nasal concha. We have 3 sets, 2 of which belong to the ethmoid bone. So we have the superior nasal concha. Actually, let me move that way over because I still need to put nasal stuff here. Sorry for your, you can't erase as easily as I can. So, cribriform plate. Nasal concha. Superior. And middle. But the inferior nasal concha, if you remember, is its own bone. Okay? It's not a process of the ethmoid bone. And the grooves under each of these conchae, called a meatus, is where our sinuses and our nasal lacrimal duct empty. Okay? One more process belongs to the nasal bone, the ethmoid bone. Anybody know what it is? Separates the right nasal cavity from the left. Time for this model. So here is the nasal bone, ethmoid bone. Here You can see superior and middle nasal concha. Here's inferior nasal concha all by itself. Here are the same concha but covered with a mucous membrane, because the purpose of that is to provide heat from venous sinuses that are close to the surface and moisture, all right, so that the air is not dry and cold as it passes down your respiratory passageways. As you look in from through the nasal cavity on our skulls, you can't see the superior nasal cavity, even on real skulls, because it's behind the middle one. So it's tucked in behind there, you can't see it, all right? So here's the cribriform, the epicrystic alley and cribriform plate, and you can see the olfactory bulb right there. Then, the other bone is the one I just took off. I mean, the other process of the ethmoid is the one I just took off. And it's this one right here. Maybe remember what we call that? Two bones contribute to the nasal septum, this lower one and this one up here. This is the ethmoid bone, but it is the perpendicular plate. So we have the horizontal palatine plates for the floor of the mouth, and then side, we have the perpendicular plate of the ethmoid. Anybody remember what this last bone is? Very thin, wide in the back, and barely visible in the front as a thin ridge. The rest of this is filled in with cartilage. Vomer, yeah, that would be the vomer. Okay, which I'm not going to draw in because it would cover up all the nasal concha, but I'm going to put in perpendicular plate. And then we have the vomer. Now, a long time ago, I read in a monthly magazine, or maybe it was a weekly magazine that came out, called Citation Index, that would put all the references for any article that was cited, and they would put in short abstracts of the articles. This is when I was in graduate school. And it said that most of you have heard the right side of the brain and the left side of the brain type of thing. And you could control which side of the brain you use during an exam by closing one of your nostrils. Unfortunately, I don't remember which, so you'll just have to try it when you're taking your exam and figure out which one makes you smarter. The other thing I read, was that you think better if you rub your nostrils and do deep knee bends. So while you're taking the exam, stand there and do this, and have somebody else punch the buttons for you, okay? I've never tried them, so I can't verify it, but you look it up somewhere on YouTube or the computer, you'll find it. All right, so much for hints on taking the exam. So as air moves through the external nares, it passes non-cornified, stratified squamous. What is the epithelium of our nasal cavity? So the space around and over the nasal concha creates the nasal cavity, and we have two of those, separated by the nasal septum. What's the respiratory epithelium? Transitional epithelium is urinary. Yes. Pseudostratified columnar epithelium. All right, so that's our respiratory epithelium. The pseudostratified columnar epithelium. So once we pass the vestibule of the nostrils, the air is going to be in contact with pseudostratified columnar epithelium. Okay? Then we leave the nasal cavity and enter into the pharynx. And pharynx is just an anatomical term for throat. So to exit that, and it becomes a narrow passageway, we end up passing through, well, we don't, but the air does, an opening known as the internal nares. That's the doorway for air to flow, or if you're getting an NG tube, for the tube to go, from your nasal cavity to your nasopharynx. And that's been really difficult, historically, for me, to get students to understand that. And it literally is a doorway, just like into the nostrils. Well, I'll put nares because it really is just one. So this model is a lifesaver for that. So we're looking at the left nasal cavity here, right? That's the left internal larynx. So it's the opening from the nasal cavity into the nasopharynx. And for the left nasal cavity, that would be that internal larynx. Now, that large head in the back of the room is another new model. We've had that one for about a year. And bring it together before it makes it hard. Let's see if I can remove this. There we go. So I'll tell you how we get this view from a cadaver. On a model, we buy it. But we have this diagram on the PowerPoints and in the lecture notes. So we're looking at posterior pharynx. And we have three regions of the pharynx. So behind the nose is nasal, behind the mouth is oral, and behind the larynx is oryngopharynx. All right? So if you look up high, there's the soft palate right there. So during lap, if you look up high, you can see the back of the nasal cavity. And you can see the nasal septum dividing the nasal cavity in half. So when you walk from the nasal cavity, not literally, when you move from the nasal cavity into the nasopharynx, you're passing through one of the internal nares. Okay? So we'll talk about the pharynx in just a moment. And this is a great model for that, because prior to that, the only concept of pharynx that we had was the sagittal head. So to identify the border of the nasopharynx, if we draw a dotted line across from the hard palate, that would be what the soft palate would do when it straightened up, as in coughing, or saying the hard G sound. So again, back to the model. This right here is the soft palate, okay? No bone structure in that. So it moves up and down, muscles that move the palate. So if we bring this up and arbitrarily draw a line, a level with the hard palate, this whole area here would be the nasopharynx, just kind of anterior to our sphenoid sinus. So we're going to have the pseudostratified columnar epithelium again, because we're not supposed to have the C sense ciliated, that's a common abbreviation. So you're not supposed to have food pass through the nasopharynx, although all of us have had some experience of coughing while we're drinking milk or soda and having it come up through our nose, okay? So we have the respiratory epithelium, pseudostratified ciliated columnar epithelia. As far as structures that we find there, we have one of our tonsils, the nasopharyngeal tonsil. All right? Just a little bit tonsil, because you already know which one it is. And I neglected to point out last night to the Monday, Wednesday students, we'll also have the opening to the internal auditory tube, all right, which that allows the middle ear to equilibrate with outside atmospheric pressure. So as we go up in an airplane or come down or go up a mountain, the air around us changes density, and that allows the connection from the middle ear to the external atmosphere, and air molecules should move. If you have a cold, it's difficult for that to happen. Yeah?
Is the nasal cavity ciliated as well?
Yes, same thing. All right? Now we're going to move into the oral pharynx. We're going to draw on the tongue. And we have our palatine tonsil is in the oral pharynx. And our lingual tonsil is in the oral pharynx. And that's going to start at the end of the nasopharynx. At the base of the tongue, you can almost touch it with your finger if you want to gag. We have the epiglottis of the larynx, okay? And we have the opening for the larynx of the cartilage plate. And then down here, we have the last cartilage of the larynx, the cricoid cartilage. So from this level to the last part of the larynx, the cricoid cartilage, is going to be our laryngopharynx. All right? So this is oral pharynx. And laryngopharynx. The epithelia of the oral pharynx and the laryngopharynx is back to the epithelia that we share for resisting abrasion. Air is a shared airway with both air and food. It's a shared pathway for both air and food. And none of us typically chew corn chips into mush. And so we have the protection of multiple layers of cells. And so we're back to the epithelia of the vestibule, which is the non-cornified stratified squamous epithelia. And that's because food passes through both of those pathways, all right? So here and here is pseudostratified columnar. When we label epithelial, we use the number of layers and the shape of the cells. It is ciliated, but I don't typically include that in the title, but PSCC is a common abbreviation for that. Then we get into the oral pharynx, and that's non-cornified stratified squamous, and laryngopharynx is also non-cornified stratified squamous. Okay? So this view of this tissue, it looks pseudostratified. It looks like we have multiple layers of nuclei in cilia, but in fact, there are nuclei down here and none up here, and this is really not even epithelium. It's neurons. This is why it looks like it's pseudostratified and why it looks like it's ciliated, all right? So that previous image is the one I will always show you. This picture right here, when I want you to identify olfactory quote-unquote epithelia, right? But it's the receptors and the neurons for smell. And that's going to be what we find immediately on the roof of the nasal cavity or at the cribriform plate. Okay? So question number 2 says olfactory epithelia is formed of olfactory neurons and supportive cells. And these neurons are classified as special visceral afferent. Visceral meaning, afferent meaning they're sensory. Visceral meaning they're sensory from an organ, not from the muscles or the skin. And special meaning they're special senses. Same thing for the neurons that we have in the eye or balance. So by shape, we classify them as pseudo-unipolar, bipolar, multipolar, or unipolar. That's a description from Bio 430. All right? Sorry, we're hitting that kind of hard today. So, here is unipolar. One branch coming off of the cell. Bipolar has 2 branches coming off the cell. So when an action potential travels, it travels along one process, around the cell body, and out the other process. That takes a bit of time, all right? So pseudo-unipolar, the bipolar neurons come together to form a stalk, and then we have the 2 branches. That creates a more efficient transmission of our nerve impulse. And if you've forgotten, this is the one that forms the dorsal root ganglia. All right, so carrying in pain, touch, and temperature from the periphery of your body, dorsal root ganglia, this goes into the spinal cord. All right? Anybody remember where we find the cell bodies of multipolar neurons? Multipolar neurons are the ones we talked about for neurophysiology with dendrites and a single axon. going out to axon terminals. Multipolar are what form the anterior gray forms of the spinal cord. Or motor, sensory. Anterior, motor, all right? So bipolar we find in only 2 locations in the body after embryonic development. The nose, there's your answer. I had to point it out. That was your answer. And the other place, anybody remember the one other place where we find bipolar neurons? Also a special visceral afferent, a special senses? Eyes in the retina. Okay? It's a little hard to see in this image that these cells are bipolar, but you can see it in the previous diagram here. Okay? So if you remember, if you had me for Bio 430, we talked about first order, second order, and third order neurons. These would be the first order. Okay? I'm not going to test you on that because not everybody had that here. But the second order are shown up there as the olfactory tract. So when we talked about in Bio 430, second order neurons were always in the spinal cord. So even though olfactory nerve is a cranial nerve, embryologically, like the optic nerve, is part of the spinal cord, and so it's affected by spinal cord infections, like meningitis or other conditions. Even though structurally we think of it as a cranial nerve. All right, so here's the respiratory epithelia. A nice plastic section, so it's nice and clear. You can see the thick basement membrane. All epithelia have a basement membrane, but it's particularly thick and pseudostratified. Why do we call it pseudostratified? It's not really stratified, which means what? It's not stratified, how many layers do we have? One. Okay. So even the nucleus up here, actually somewhere in front of this slide or behind the slide is going to come down to the basement membrane. All right. What else do we always see in pseudostratified in the respiratory tract at least? Cilia. Okay. Now that's where I left it when I taught Bio 430 last semester. But earlier this year we learned that pseudostratified in the male reproductive tract has microvilli. Okay. But for the respiratory system, it's always going to be cilia. There might be some microvilli, a few microvilli, but primarily it's cilia. Okay. So while you answer the third question, may I erase the board? You guys done with that? We're done with nasal cavity and pharynx, except I'm going to draw the pharyngeal muscles. So here's the third question. And doing my daily search for the eraser. Right. Okay, that's what that was. But I see my eraser.
I think it might be under your
Holding it up.
What? I think
It's been 20 minutes rather than under the hammer.
Do they all touch the base of the membrane? They're supposed to.
I think they're supposed to.
So before we talk about the cartilages of the larynx, let's talk about the throat, all right, the pharynx. So sore throat would be an upper or lower respiratory tract condition. Upper. Now pharyngeal muscles that are called constrictors, or the complete pharyngeal constrictors, right? They're primarily posterior. They're open laterally. And the deepest one is the superior, and the middle one overlaps that, and the inferior one overlaps that, all right? There are two sets, one on the right and one on the left. Actually, this would be right, this would be left. And they meet in the middle in a seam-like region called a raphe. We saw that with the bulbospongiosus muscle in the male, all right? We saw that in the scrotum as well. So this would be the raphe right here. So here's the superior, middle overlaps that, and the inferior overlaps that. If we go back to the previous image, okay, you can see that a little bit more clearly. So here's the superior, middle, and inferior. And then the inferior blends in with the upper skeletal muscles of the esophagus. So these are going to be used when you first start swallowing. The superior has this attachment to the buccinator muscle. Remember the buccinator? Facial or chewy? Facial. It's the one you use to suck in your cheeks, compress your cheeks, and we'll look at it when we talk about the digestive system, because it keeps food in your oral cavity. So that's the attachment for the superior constrictor, all right, up here. The middle constrictor comes off of the hyoid bone, and the inferior constrictor attaches from the cricoid cartilage, the most inferior cartilage of the larynx, and thyroid cartilage, okay? And then blends in with the esophagus, which is complete, its own structure. It's not attached to the trachea, all right, and forms that component. Now, that model that you see over there on the table is a model that I'm grateful to get, because we haven't done that dissection yet on any of our cadavers. It's actually one of my favorite dissections to do on a cadaver, because of what you can see from the posterior view. You can dissect, you can find the glossopharyngeal nerves, you can find the hypoglossal nerves, all in this back area of the throat, and it's just a cool view of the posterior region of the cadaver. So, to do so, if you, and don't do this if you have long fingernails, all right, mine are medium and it's gonna hurt, but the trachea lies anterior to the esophagus. The esophagus lies anterior to the bodies of the cervical vertebrae. And I wouldn't do it with the skin, but you, and then someone who's died and you've taken the skin off, you can actually put your fingers behind the esophagus and push them through, and they can touch, you can slide them up and down. It's a passageway actually for infections to travel back through there, and you can pull them away from the cervical vertebra and create a space there. Slide that up and free it, and then cut the ligaments of the skull, the occipital condyles and all around, from the first cervical vertebra, from the atlas. When that is all done, then you can take the head, just like the model over there, lift it up off the spinal cord, bring it forward and lay it face down on the chest. And then the view that you get is this view. And that's the model that we have over there that gives you that posterior view. Okay? So here would be the vomer bone. This would be an internal nares. This would be another internal nares. You can barely see the superior nasal concha. That would be middle, and that would be inferior. All right? So that's inside the nasal cavity. My hand right here would be in the nasopharynx. Then this is the soft palate. Here's the lingual tonsil on the top of the epiglottis. So my hand right here is in the oral pharynx. And then here's the epiglottis, the cricoid cartilage, and a few other cartilages we'll talk about with the larynx. So my hand right here is in the laryngopharynx. And then down here, behind this wall, would be the esophagus, all right, where we actually have complete separation, hopefully, between food and air. What do you think is the nerve that innervates this whole region, both sensory and motor? Not quite. Small parts, but not the whole area. Vagus. Vagus is the nerve of the larynx and most of the pharynx. All right? There's even a tiny little part of your eardrum that's vagus. So which of the following muscles of the pharynx, the constriction muscles, has its origin from the hyoid bone? I know you've seen this before, but I think maybe I didn't have time to change it. Yeah. You grew up a daycare. They grew up over most of the day. You might want to go back to this diagram for help. Okay, the answer? Help on the answer? No, I'm just saying, if you want to double check, the answer's on that image. Okay, back to your answer choices. Okay. So let's take a look at the larynx. And we're going to look at the structures of the muscle, and then we'll take a break, okay? We have 3 or 4 different models of the larynx. This is not the one I... Yep. So the view of the larynx is shown from an anterior view. This would be the trachea right here. All right, so that's not part of the larynx. So we have our hyoid cartilaginous ring. We have the anterior view. We have the posterior view. And we have an internal lateral view. All right. I'm going to do the external lateral view, but I'll use dotted lines to illustrate what we're looking at. The larynx is made up of hyaline cartilage, with the exception of the epiglottis and the cuneiform and part of the corniculate, but the majority, the larger structures, the thyroid cartilage and cricoid and arytenoids, are made of hyaline cartilage, right? As is the rest of the respiratory tract. Purpose is to provide, like we see with the trachea, a patent or open airway. It would take a lot of energy if we had to open this up with muscular force to push air out or allow air in. It also provides structural support for muscles. So the muscles that attach to the larynx modulate our voice, okay? Make it higher or lower or louder or softer. So if we look at the cartilages, we'll look at an anterior view first. Well, we'll start with one. And then we'll do a posterior view. And a lateral. So I'll draw each cartilage in sequence in each of these views. All right? So the first one is the largest, and it's the thyroid cartilage. All right? In men with the secretion of testosterone, not only do they have more dense bones and more muscle, and therefore a higher hemoglobin, they have a more prominent laryngeal prominence. Okay? So when men swallow, it's easier to see their Adam's apple move up and down. Because this, you can see from this side, this sticks out of it. All right? And that's the thyroid cartilage that is doing that. In fact, it's such a prominent male structure that individuals that are changing from male to female, if they can afford it, the hormones aren't going to change that, have surgery to shave that area down so that that thyroid is not so prominent. So this is basically an anterior cartilage. There are 2 columns that extend up posteriorly. Other than that, it's completely open in the back, okay? So there is no the U-shaped cartilage. So if we look at the posterior view, there are these 2 columns. And then we have the component in the front, all right? And laterally, posterior to the thyroid cartilage is the cricoid cartilage. That's narrow in the front and wide enough in the back to fill in the posterior wall that was absent by the thyroid cartilage. So I'll draw that one in blue. And there's a ligament that connects the cricoid to the thyroid. And there's a ligament that connects the thyroid to the hyoid bone. Okay. So up here, this is called the thyrohyoid ligament. Sometimes biology makes sense. It's not when we're naming clotting factors. So posteriorly, I don't want to go that high because I'm some other cartilages in there. We have the cricoid. All right, so from the posterior view, resting on top of the cricoid, we have two pyramidal shaped cartilages. I can't draw three-dimensional pyramidal on the board, so I'll draw two-dimensional. And we'll just put A. All right, and these are the arytenoid or arytenoid, depending on how you want to pronounce them. Arytenoid may help you remember how to spell it. Arytenoid is how they're most often pronounced. And that's these two right here. Okay? They swing on an axis that allows them to change whether or not the ligaments are apart or close together. So those are the major hyaline cartilages. Then sitting on top of the arytenoid, we have a small, another kind of pyramidal cartilage called corniculate. And it always reminds me of a kernel of corn. So that helps me remember that one. Okay. And the next one is very thin. Essentially, I'll draw it out here because I can't draw all of it in there. Essentially, it looks like this. This top part is about the size of a large straight pin head. Not the small sewing ones, but the bigger ones. All right? And it has a very slender stalk. And this is elastic. All right? And that kind of comes up the side here. Okay? The one that I'm not going to draw on there, well, I guess I could put it here, is the epiglottic cartilage. It's attached to the inside of the thyroid right here. And it's a leaf-like cartilage that comes down over the opening of the larynx to block airflow. And this is epiglottic. Epiglottis is what we call the whole thing with the rest of the soft tissue, the membrane, and everything else. But the cartilage itself is the epiglottic cartilage. All All right, so I'll show you where else we can see the cuneiform. So you can see on our cartilage models everything but the cuneiform. All right. So here we have thyroid bone. So this is showing the thyrohyoid muscle, which we're not going to talk about, but the space in here would be filled in with the thyrohyoid ligament. Okay. Then here we have the cricoid cartilage, and this would be the cricothyroid ligament. So thyrohyoid ligament, cricothyroid ligament. And then this would all be the thyroid cartilage with those two long columns in the back going up to connect with the hyoid bone. Here would be the wide portion of the cricoid cartilage. The narrow portion is anterior, okay? Sitting on top of the cricoid cartilage, we have the arytenoid cartilages. And the tiny little points that you see on them would be the corniculum, right? And we don't have the cuneiform shown here, and here would be the epiglottic, okay? All right, so let me show you a model of a small, what we call it our small larynx model. It's this one. So this is over there, and a couple of them, I believe, in a box. It comes with a tongue model as well. I'm not sure if the tongue is in there. So can you figure out which is anterior and which is posterior? Here's the epiglottic cartilage, all right? That's going to be anterior because it attaches to the inside of the anterior aspect of the thyroid cartilage. So that's our epiglottic cartilage. That makes this the thyroid. But be careful, this is not the thyroid in the back, because remember the thyroid is completely missing anything in the back. So here is our narrow cricoid anteriorly, and here is the wide portion of the cricoid posteriorly. So we can't see the arytenoid cartilage because we have muscles that connect the two arytenoids, and when we cut in the midline, we're cutting through those muscles. But it has that shape of the arytenoid, so just a few millimeters in would be where we should find the arytenoid. Obviously, this is a model, so a few millimeters in is going to be plastic, but that's where it should be. This bump right here is due to the corniculate, and then you can see that there is a membrane. It connects from the arytenoid cartilage up to the epiglottis. You don't have to know this membrane, but I'm talking about it. It's called the arytenoid epiglottic membrane. You might as well, because I got to test you on it. Certain African tribes and maybe other countries, I'm just not aware of them, speak through additional speech and vibration of the vocal ligaments, and they use this epiglottic fold to vibrate it and give additional sounds. Okay? I don't know if you ever saw, it's an old movie, probably 35 years old, called The Gods Must Be Crazy. There's a God Must Be Crazy too. It's hilarious. But their illustration of that kind of vocalization is in that movie. So this bump that you see right here and right there is due to that larger head of the cuneiform cartilage. That's the only way we're going to see it in any of our models, okay, is that bump. And I usually mark it with a pen. So if you see a picture of this model with the pen at that point, I'm looking for cuneiform cartilage. While we're on this, you can see that there are two edges or folds here. These are for the vocal folds, or inside the vocal cords or vocal ligaments. The upper one is the false, also known as vestibular, and the lower one is the true, also known as ventricular. So if you're talking or writing an answer about the vocal cords, I want you to use false and true, not just vocal cords, okay? So that I know you know whether it's false or true. So typically true are just called vocal cords, but I'm going to ask you to add the word true so that I know for sure you know it's true, all right? Or you can call it ventricular if you wish. All right, so let's test your short-term memory. Which of those cartilages is indicated by that bump right there? So it's this one right here that I have. Okay. So we have, these are the arytenoid muscles, if that gives you a clue. And then I'm sitting on top of them is one cartilage. Up that area epiglottic membrane, we have the other one, is what I have. Okay. And I didn't write it on the board. You're going to have to find it on the answers. It's also the name of three bones in the Thursday, yeah, it's not in August, so you do need to know.
You said just some notes?
Down to the larynx. From that picture, you can see the larynx. Well, you wouldn't have anything to ask.
That's what I was wondering. So how about that visual space?
Well, I have to come up with 4 different sets of 6 questions. So that, the cartilage and the muscles of the cartilages of the larynx and the muscles of the larynx is where we'll stop. What you need to know for this, okay? Anybody need more time on this image? Okay. So, when we look at this diagram, all right, here would be the false vocal cord forming the vestibular fold, and the true vocal cord forming the ventricular fold, and right in here would be the actual collagen fibers forming the true vocal ligament, and around it would be the muscles that surround the vocal ligament. All right? So we're going to look at those muscles next, and let's start with this one. Okay, and then we'll take a break. Come back and watch some videos of the larynx. So in your lecture notes, there's a diagram that shows these. Two diagrams, and those are these diagrams and this diagram. Okay? However, there is an error on page 19 down where it talks about arytenoids. It says they AB duct, they actually AD duct. Okay, there's only one muscle that's AB duct. So on page 19, where it talks about the arytenoids, change that from AB duct to AD duct. AB, they bring together instead of taking apart. All right, so let's look at the muscles. And you have them written in your lecture notes, so I'm not going to erase this yet to talk about them, but let's show them on the model. All right?
Not ADD.
For which one?
So cricothyroid is this one right here. It connects the cricoid cartilage to the thyroid. The point of attachment that we call insertion, the movable attachment, is the thyroid cartilage. So when the cricothyroid contracts, it tilts the thyroid cartilage forward and away from the arytenoid. The vocal ligaments are attached between the arytenoid and thyroid. All right? So when we tilt the thyroid cartilage forward, we are stretching the vocal ligaments. That's going to change the pitch of your voice. Is it going to make it higher or lower? Higher. So if you've got a guitar or a string bass or whatever, and you're turning the tuning pegs, when you tighten them up, the pitch goes up. Okay? That means you go a little higher like that. And that's what cricothyroid does. It tilts that thyroid cartilage and stretches out the vocal ligaments. Okay? So that would be cricothyroid. Posterior cricoarytenoid is the only one that actually AB ducts, and it's this muscle right here in the back. Let me show you a better picture. Here. This one right here. Okay? So it starts off the cricoid and wraps around the side to the arytenoids. It used to be called posterior cricothyroid, which was weird because it doesn't attach to the thyroid cartilage. Okay? And what it does is it does this. It pulls apart, or AB ducts, the vocal ligaments. So this muscle right here, posterior cricoarytenoid, is going to do this movement here. So it turns the arytenoid cartilages and increases the space between the vocal ligaments, and that makes the voice louder. You can send more air through. Okay? So that's the posterior cricoarytenoid. The lateral cricoarytenoids, you probably need to look at this model because you have to look down the side. And on the side of the cricoid cartilage, on the inside, where it's wrapped around by the thyroid cartilage, is our lateral cricoarytenoid. Okay? So on the inside of that, so that's this one here. And it's going to AD duct. It's going to bring the vocal ligaments together. And And then the arytenoids, also adduct, we have two sets of those. These are the ones that we saw in the sagittal section, so going back and forth, this is like a barn gate. So going back and forth, straight back and forth are the transverse arytenoids. And superficial to those, we have a crisscross, and those are the oblique. All right, so those are seen right here. Going from arytenoid to arytenoid. Transverse and obliques, both are going to bring the vocal ligaments together. And finally, the last one I want you to know is inside. Not the most lateral one here, but the most internal one, right around over the vocal ligaments, called vocalis. That is the antagonist to our cricoarytenoid. So the cricoarytenoid pulled the thyroid out and stretched the vocal ligaments, making your pitch higher. Vocalis takes a hold of the thyroid cartilage and pulls it back towards the arytenoid, makes the vocal ligament shorter, and therefore the pitch is lower. Okay, let's go ahead and stop there and take a break. When we come back, we'll see a couple of videos of those working, okay? And we'll talk about the nerve, the innervates, the one nerve that we break into 3 different names. It innervates all these muscles, and then we'll move into the airways. So why don't you come back